In this video, we will discuss about some of the sources of NK92 cells and T cells that can be used for the clinical trials. So, for this, we need to take uh, two examples or samples from two different persons. One is the healthy donor and another one is the patient. For NK cells, there are several approaches available. That is, NK cell lines can directly be used for the clinical trials, but after irradiations, so that there is no like undesired mutations will occur in the NK cell lines, and this NK cells can then be this CAR gene can be transduced, and this CAR NK cells can be used for or infused into the patients. Apart from the direct cell lines, it can also be produced using induced pluripotent stem cells through the differentiations and through this ubiquitous blood can also be used. From that ubiquitous blood, from that NK cells can be isolated, and from the PBMCs also, or these PBMCs can be taken from the patients or from the healthy donors. When it is taken from the patients, it is usually called allogenic, and it will have less side effects or high circulation times, or we can say high half-life period if NK cells are taken from the autologous from the patients itself. And once this PBMC has been extracted from this healthy donor or patients, then the NK cells can be isolated from them. And from the iPSC, it can directly be differentiate first into the NK cells, or what they can do is they first transduce this CAR gene into the iPSC. That is, then they form this CAR iPSC, then the differentiation into the CAR NK cells, and then infused into the patients. But for the T cells, there are not very much options available. One of the common source for the T cells is patient itself, or in the form of autologous. That is, autologous T cells. That is, first the PBMCs are extracted from the patients, and then T cells are isolated. Then these T cells will undergo CAR gene transductions, and these CAR T cells then be infused back to the patients. But if the T cells are taken from this healthy donor, then in that case the T cells that are isolated from the PBMCs may cause allogenic T cells, may cause GVHD, or we can say that the immune systems of the patients will respond to these T cells, as these are the T cells from the different persons, not the autologous patients T cells. So in general, the autologous T cells are being used for these CAR T cell therapies, but for the NK CAR NK therapies, there are many options for the NK cells can be used, and we have discussed in the previous videos that different types of or different generations of this CAR has been developed to trigger different signaling pathways. It is co-stimulatory. In the second From the second generation, the co-stimulatory domains are also been added, so that it can not only triggers the formations of the cytokines such as Granzyme or perforin, but it will also cause the activations of the host immune systems, and thereby having the cascading effect. So we can say that this CAR immunotherapy's main function is to activate the immune systems or prime the immune systems of the patients, so that the patient's immune systems will be able to work against the tumors or the tumor associated antigens. That is, B cells can be able to generate antibodies that is specific to those antigens, and T cells will be able to release cytokines that helps the tumor apoptosis, and these cycles will continue along with this CAR cells functions, and this thereby it helps in the treatment of the patients who are affected with these tumors. So that's all for this video. If you need to understand more about this topic, the resources of NK, NK2 cells and T cells for CAR immunotherapy, you can go through the reference. Go through the reference that has been provided in the description. Thank you for your time.